Hi, and welcome to the iBio lecture called Chemical Glycobiology. My name is Carolyn Bertozzi, and I'm a professor of chemistry and molecular and cell biology at the University of California at Berkeley, and also an investigator of the Howard Hughes Medical Institute. I am a chemist by training who became interested in the biology of sugars, which is what the term glycobiology means, when I was in graduate school and then later as a postdoctoral fellow. And my laboratory research at UC Berkeley seeks to combine chemistry and biology together to understand what sugars are doing in the human body. So let me begin by giving you an introduction as to why I'm so interested in the biology of sugars, along with all of the students and postdoctoral fellows that work with me in the laboratory. It turns out that around the turn of the millennium, there was a very exciting sequence of breakthroughs in biology having to do with sequencing genomes. So in the early days of genome sequencing, the first eukaryotic organism to be characterized in this way was the budding yeast. And one of the great surprises from the sequence of the yeast genome was the fact that it only contains about 6,000 genes. And that's not a very large number. In fact, before the genome was completed, there were some estimates that there would be far more genes required to encode all of the interesting functions that eukaryotes perform. So the number 6,000 seemed very small. Of course, the budding yeast is a relatively simple eukaryotic organism. It's a single-celled organism. And there was this idea that more cells would require more genes. And that seemed to be the case as more and more genomes were decoded. So when the C. elegans genome was finally sequenced, that genome had about 15,000 genes, which is a larger number of genes, and it seemed to make sense. And then the next major organism to have its genome sequenced was Drosophila, or the simple fruit fly, which had about 20,000 genes. And all the while, scientists were working on the human genome, operating under the assumption that the human genome would be much larger than any of these other model organisms. In fact, some early scientists estimated that the human genome might have over 100,000 genes. Well, around the turn of the millennium, there was a big surprise yet again in the world of genome sequencing when the human genome was finally completed, and it turns out that it's not that much larger than the fly genome or the C. elegans genome, and really not even that much larger than the yeast genome. There are only around 25,000 genes encoded in the human genome. These are the genes that encode for proteins. And by the way, I think that nobody would be more surprised than Charles Darwin himself to discover that we really don't have that many genes. So then the next question became, given that we only have about 25,000 genes, how is it that human beings can be so biologically complicated compared to some of these other simple model organisms? Well, in one of the major publications in 2001 that put forth the sequence of the human genome, Craig Venter and his colleagues made this statement, which is that the finding that the human genome contains fewer genes than previously predicted might be compensated for by combinatorial diversity generated at the level of post-translational modification of proteins. So, put another way, what happens in the higher, more complicated organisms is that the proteins encoded by the genome are modified in very complicated and diverse ways to create a lot more biological complexity than one might predict just by simply counting the genes in the genome. And modifications of those proteins are what we call the post-translational modifications. Well, it turns out that one of the most complicated of those post-translational modifications is the attachment of sugars to those proteins. And that's the process that we call glycosylation. And by the way, you'll see that prefix glyco again and again and again throughout these lectures. That's the Greek prefix for sugar. It turns out that many of the proteins that are glycosylated are proteins attached to the membranes of cells. In fact, they reside on the surface of cells, and we call those sugar-modified proteins glycoproteins. And an example of one of those glycoproteins is shown here in cartoon form, where the protein is this blue structure that's anchored in the membrane of the cell. We also have lipids in our cell membranes that have sugars attached to them. So for example, this cartoon illustrates what we call a glycolipid. And the sugar part of the glycoprotein or the glycolipid is what we might refer to scientifically as a glycan. Glycan is just another word for a complex sugar molecule. So because of all of these glycoproteins and glycolipids on the surface of our cells, we basically can think of our cells as having a sugar coating. And in fact, that's why on the very first slide of this lecture, I had a cartoon of M&Ms. Because in many ways, you can think of our cells as being like M&Ms. They're coated with sugar molecules. Now, that to me is fascinating. And of course, it begs the question, what are the functions of all of these sugar molecules that are attached to the proteins and the lipids and decorating the surfaces of our cells? And that question, what are the functions of the sugars? That is the question that's embodied by the field of glycobiology.